The overall goal of this protocol is to develop and characterize murine tolerogenic dendritic cells for the evaluation of their immunotherapeutic utility in the treatment of autoimmune disorders such as multiple sclerosis.
This method can help answer key questions in the tolerogenic dendritic cell therapy field by providing a standardized method for developing and functionally characterizing tolerogenic dendritic cells. The main advantages of this technique are that it can be used for successful tolerogenic dendritic cell development and for functional tolerogenic dendritic cell efficacy testing.
The implications of this technique are significant. They extend toward the development of cellular immunotherapy for autoimmune disorders, as tolerogenic dendritic cells can reset an abnormal immune response while remaining subject to intrinsic immune regulatory mechanisms.
After harvesting the hind leg bones from 8 to 10-week-old C57BL6 mice according to standard protocols, use surgical blades and forceps to dissect as much tissue as possible and place the clean tibias and femurs in a 6 cm culture dish containing 70% ethanol. Trim both ends of the bones with a surgical blade and use a 3 ml syringe equipped with a 23 gauge needle to flush the marrow from the first bone with 3 ml of PBS into a 15 ml conical tube. When all of the marrow has been collected, centrifuge the cell suspension and resuspend the pellet in 1 ml of red blood cell lysis buffer. After 5 minutes, stop the lysis with 9 ml of PBS and collect the cells by centrifugation. Resuspend the white